In this lab, I performed the desalting protein and affinity chromatography techniques. The column is first equilibrated with 3 ppm SF buffer through multiple centrifugation washes. Meanwhile, the protein pellet is fully resuspended in 3 ppm SF buffer and kept cold to maintain stability. The sample is then loaded onto the column, where desalting occurs and the purified protein element is collected and stored for further analysis. Following, the desalted sample is loaded onto the affinity column, and fractions are collected as buffer washes remove unbound proteins, and NADH wash is then used to competitively elute the bound protein by disrupting its interaction with the ligand. The purified fractions are identified using absorbance at 280 nanometers and stored for further use. The sieve spin column effectively removes salt and small molecules such as ammonium sulfate while preserving the protein of interest. The biochemistry behind this technique is based on size exclusion chromatography. The column contains a porous resin that traps small molecules like salts inside its pore, while larger molecules such as proteins cannot enter and instead pass quickly through the column during centrifugation. This allows for efficient separation based on molecular size. In affinity chromatography, proteins are purified based on specific binding interactions. Its purpose is to isolate the target protein by exploring its unique chemical affinity for a ligand attached to the column. Biochemically, this method works through a highly specific interaction between the protein and the immobilized ligand, in this case cytochrome blue, which mimics nucleotide cofactors like NADH and binds proteins with nucleotide binding sites. Non-binding proteins are washed away using buffer while the target protein is retained.